So in lecture, we started talking about different things that microbes need to grow and be happy and reproduce. And a lot of that is different elements right off the periodic table, such as carbon, such as hydrogen, oxygen, and nitrogen. Those are really the top 4 elements that organisms need for survival. These are the top 4 elements that humans, not only just humans, as bacteria and most organisms need for survival. So we briefly talked a little bit about carbon and how they can make their own carbon, how they can get their carbon from food sources if they're heterotrophs. And then we also talked a little bit about the hydrogen. And when I say a little bit, the only time I talk about hydrogen and how they get it is right here at the bottom of the slide. But oxygen is another one of the big requirements that organisms need. Now, how does oxygen affect different organisms? Well, when you see the word obligate, it's really saying required. So if you're an obligate aerobe, that means you require oxygen. If you're an obligate anaerobe, it means you require no oxygen. But the problem is organisms that are aerobes, they require oxygen, they need oxygen in various chemical formulas. It's those chemical formulas, the different reactions that occur in the cell, can take oxygen, normal oxygen, O2 in the air, and turn it into a toxic form. So toxic forms of oxygen form from various types of just metabolic reactions, chemical reactions in the cell. And toxic forms of oxygen are highly reactive, which means they are constantly trying to do things even when they're not supposed to be doing them. They're also excellent oxidizing agents, which means they're losing electrons. And electrons, they're just kind of free range out there in the cell. And when you have these kind of free range electrons, they can damage the cell. They bounce around, damaging different structures. Now, different examples of toxic forms of oxygen are listed in this table down on the bottom. If you have a single oxygen molecule all by itself, because oxygen is always happy and stable when it's in a pair. But if you have a single oxygen, it's not happy, it's not stable. A superoxide radical, which means it just has an extra electron, again, it's energized with that extra electron, or a peroxide anion has 2 extra electrons, meaning it is doubly reactive and energized. Now, if there are superoxide radicals that have these extra electrons around, how do cells fix that? Well, some cells make enzyme called superoxide dismutase. It's an enzyme, ends in an ase, and it can take this toxic form of oxygen and neutralize it. Same thing, peroxide. It has 2 extra electrons. Organisms can make enzymes, catalase, peroxidase. We're going to work with catalase in the lab, testing if organisms make it. And these enzymes can neutralize that peroxide and make it non-toxic, non-reactive. That single oxygen, different organisms have pigments called carotenoids that can neutralize it. So if an organism requires oxygen, they have to know that there are going to be toxic forms of oxygen somewhere occurring during different processes, during different chemical reactions, that organisms that require oxygen have to somehow have a way to detoxify them. Now, if you're an obligate anaerobe, which means you cannot survive in oxygen, you require no oxygen, it's because they don't make any of these enzymes. Like, they cannot survive in an oxygen environment because the toxic forms of oxygen, if they're exposed to oxygen, it will form toxic forms and it will kill them. So an obligate anaerobe has to live in a zero oxygen environment because those toxic forms of oxygen will kill them. So how much oxygen can an organism require? So if something is an aerobe, it means that they want oxygen. And if we try to grow some of these just strict aerobes in a lab, if we put them in an auger, but we put it in a tube, they're only going to grow in the auger near the top where the highest amount of oxygen is going to be found. If they're an obligate anaerobe, that means they cannot survive anywhere where there's oxygen. So they're not going to grow anywhere near the top of the media where there's oxygen present. They're only going to grow in that farthest anaerobic area of the tube. But there's kind of some of these in-betweens. There can be something known as facultative anaerobe. So a facultative anaerobe, it's like the word faculty, I always think they can do it all. They can grow with oxygen, but they can also grow without oxygen. Oxygen will not kill them. They're going to grow best and happiest where there's oxygen present, but they can still survive a no oxygen environment. So it's like they can grow in both. They'd rather have oxygen, but they can grow without oxygen as well. Then there's something known as aerotolerant, and aero is referring to oxygen. Means they don't require oxygen for any kind of chemical formula, but it won't kill them either. So they just tolerate oxygen. So they're not happiest growing with, if oxygen is present, they really just don't care. So they can survive in no oxygen environments, but they can survive in aerobic environments as well. Really, they just tolerate oxygen, they just don't use it. So you can find that uniformly in a tube. And then there's also something called a microaerophile, and we'll work with some of these in lab this semester. This "phil" part of the word means it likes. Micro means small, aero means oxygen. There are some organisms that like small amounts of oxygen. So they grow best where there's some oxygen, but not a lot of oxygen. I don't have a picture on these tubes, but if they were going to show it, they would show bacteria growing kind of in the middle. Not where there's lots of oxygen, but not where there's no oxygen, kind of somewhere in the middle. This is showing an example of where you can look to see where are bacteria growing to kind of give us a clue. Are they growing uniformly throughout, only near the top, only at the bottom? It gives us that clue on what kind of oxygen requirement that organism needs, and then also is a clue on what the organism is. Now, some other elements that that bacteria need is nitrogen, and a lot of bacteria can do what's known as nitrogen fixation, which means they can take unusable nitrogen, such as nitrogen gas that's in the air all around us, and they can turn it into a usable form like ammonia. Ammonia is used in a lot of chemical reactions for building up of other molecules, but nitrogen gas that's in the air we're breathing right now, that's not a usable form. It can't be used in any chemical reactions. It's not reactive. It's very stable. It doesn't really want to do anything. But bacteria are known for doing nitrogen fixation. So they're taking unusable forms of nitrogen and turning them into usable forms. What's worked out really well is a lot of plants need nitrogen as well, and they've actually formed relationships with bacteria. So this is showing bacteria on and attached to different plant roots, because plants need nitrogen, but they can't do nitrogen fixation. They can't take nitrogen gas that's in the air around us and turn it into a usable form, but they'll form an association with bacteria that can, so that these bacteria are like little instant fertilizers that are giving these plants the nitrogen that they need. Phosphorus and sulfur are also another couple of the elements that bacteria need and thrive on. Trace elements, again, there's lots of elements off the periodic table, whether it's calcium or another element that, yes, they usually need these trace elements, but they're not the big elements they need for growth. And then there's something known as growth factors. These are necessary organic chemicals. So a larger molecule that's just not an element, some type of organic chemical that an organism can't make, but they're needed for survival. Oops. Oh, I thought I had more written on that slide. There's different types of heme. The heme molecule itself, it's an organic molecule. Cells can't make that, but they do require it. So if we have to grow an organism that requires these different growth factors, we just have to make sure we add that to the plates. Other requirements is different temperatures, and that's just because certain temperatures affect different areas of the cell. So if you heat up a protein, you denature it. As I mentioned in lecture, that proteins work at best at different temperatures. That's because they have a very specific shape. If this was the yarn that knitted your sweater, if this was knitted into a perfect sweater, it would do its job of keeping you warm. But if you unwound it and just had a big pile of yarn, it's not going to do its job anymore. And if you heat heat a protein up, it will unravel it and it can no longer do its job. Also, the cell membrane, whether it's a bacteria, a prokaryotic cell, or a eukaryotic cell, is a phospholipid bilayer. And that lipid, if you've ever heated up any kind of lipid fatty substance, it melts. Same thing with butter, is a very lipid molecule. And if you heat it up, it melts, and that changes its whole shape and whole structure. And that would be bad if you melted the cell membrane. This is showing that different bacteria, again, work best at different environments. And a lot of that is just because if you go outside of those environments, proteins don't work as well, and you might start causing damage. Doesn't mean they don't always work or grow. It's just if you go too far outside of their optimum, then you can start causing damage. This is showing, this is a bacteria that's going to grow best at body temperature. But if we had it, say, just sitting on the counter, that doesn't mean the bacteria won't grow, or even in a refrigerator, but they're growing a lot slower. They're not growing at their optimal growth rate. We, again, can group organisms based on what their optimal temperatures are. So generally the ones that grow in us, so those are the ones we focus on in this class, they're called mesophiles. Meso kind of just means middle temperatures. They're not too hot, they're not too cold. And that phil or phile in the name means it likes. So it likes kind of middle-ish temperatures. And so you can see its optimal growth temperature really is right around body temperature. But there are some organisms out there, again, they generally don't cause too much for human diseases, so we don't care about them too much, that like really warm temperatures, even above boiling, such as those found in hot springs. There are some organisms that really like cold temperatures. This picture, I've got a zoomed in larger picture of it. This is an organism that grows on cold temperatures. It's an algae it's a red pigmented algae that actually grows right on top of snow. It grows best in cold temperatures. I like the prefix, the fact they've called them psychrophiles, because I always think, well, you're a psycho if you're liking these cold temperatures, as we're going to get come winter time. Also, there's another physical requirement organisms need, and that's they have to have a certain pH for them to grow well. If you have too many hydrogen ions in an acid environment or too many hydroxide ions in an alkaline environment, it interferes with hydrogen bonding. It interferes with making and breaking down different molecules. Molecules are not going to be able to grow and divide. So microbes that are associated with humans generally prefer a more neutral pH, right around that 7.3 to 7.5, closer to that blood pH. But there are some organisms that can still tolerate and survive a pH above or below that neutral. So if an organism likes a neutral pH, we call it a neutrophile. Again, that phil or phile means it likes, so it likes a neutral pH. An acidophile means it likes acid. There are bacteria that can survive and grow in your stomach, a very acidic environment. Doesn't all mean they love the acid, but they can tolerate it. There are some bacteria that live in your intestines, which is an alkaline environment, especially in your small intestine. Again, they grow and thrive wonderful in that environment. And again, doesn't always mean that they really like it, even though it does have that phile suffix in it, but they can definitely tolerate those pHs. Another physical requirement that microbes need is water. Just like us, we require water. Main reason why water is so important, it's a universal solvent. It's going to dissolve almost anything. This includes different nutrients, different enzymes. It's also important in metabolic reactions. A lot of times water is needed to break things apart. We even saw that in the Krebs cycle. Water has to go in part of the process for breaking down different molecules. So what happens to cells with no water? If you require water and now you don't have water, you're not going to survive. But some cells still survive in dry conditions, at least for short periods of time. It depends on the organism. They have a lot of mechanisms so that they can survive a dry period. So that a bacteria sitting on a table, maybe right next to you, is sitting there and it's not dying yet. It may be slowly dying if it doesn't ever get water or end up on another more habitable surface. But it has a cell wall that tries to really hold in the water. It may have two membranes like a gram-negative bacteria. It may have some type of lipid covering found in its cell wall, all to try to keep that water in. And then there are some organisms, if you're in the Bacillus or the Clostridium genus, that can make endospores and just go dormant if there's no water around. And then there's some bacteria that require a very specific osmotic pressure. So when we're talking about osmosis, we're talking about moving of water to where there's not as much water. So we're talking about, if we've got microbes living in different types of habitats that can change the flow of water in or out of them, they have to be able to adapt to that. So a halophile, again, that phile, it means it likes something. Halo means salt. There are still microorganisms that are living in the Dead Sea. We figured out it's not dead because it's so salty. We were under the impression nothing could survive in that high of salt. There's bacteria that can. There are bacteria that live in the Great Salt Lake. Again, it's a very high salt content. Now, generally, if you put any kind of cell into a very salty environment, water wants to go where there's not as much water, and it will leave the cells. The cells will shrivel up and die. Well, some bacteria have figured out ways of surviving that, and even sometimes thriving, that if you took them out of the Great Salt Lake and put them in fresh water, and I'm like, they would literally explode. The water would go in and they would explode. And then there's some bacteria that require a specific hydrostatic pressure. Now, I love that I put this little clip in because it takes you back to, if you've seen the movie Finding Nemo, which I hope you have, of where were they when they were encountering this particular fish. They were down in the great deep crevices at the very bottom, the big trenches. And when you get down to the bottom of the ocean, especially those deep trenches, there is so much water pressure. Well, there are bacteria that are down there that can survive that water pressure. There's fish that can survive it too. And I'm like, most organisms, if we went down there, we would get squished. But there are some bacteria that have survived and adapted to that high water pressure. Now, moving away from some of those physical requirements, because there are some organisms that have adapted to always live with other organisms. Whether they cause them harm or not, they have adapted to live with different organisms. So organisms that live in association with, we're just talking about organisms that live in association with some other different species. Some of them are non-symbiotic relationships, which means they're not all benefiting from it, and some are symbiotic relationships, which means I want to say someone's benefiting from that particular association, but it is a required association. So a symbiotic relationship, which means one of the organism, it's required that they live in this type of environment, whether it's because they're always causing harm or because they're always benefiting. Some different examples: mutualism means both organisms are benefiting from it. Commensal organisms means just one organism benefits from the relationship, but the other one's unaffected whatsoever. Again, you probably didn't even realize how many bacteria you had on your skin until we did the hand washing. Commensal relationship. They were benefiting because you gave them a home. I'm like, but you were unaffected by those normal flora. Parasitism means they're going to benefit at your expense. Now, biofilms is a non-symbiotic relationship. It means the organisms don't have to live in this relationship, but in a biofilm case, they're going to benefit from it. And so a biofilm just means a synergistic relationship among numerous microorganisms. So microorganisms that are all going to live together, and in a biofilm, they're going to benefit from it. Again, bio means living, film means a covering. So these are bacteria that are going to live together and they're going to cover a surface. Now, because I've got several things about biofilms, the benefits of organisms all living together is that they can help cells attach to one another. Bacteria can't grow until they first find a home. So by having one bacteria stick, maybe it leads another bacteria to stick. It allows them to attach to surfaces themselves. It allows them to then sometimes all work together to get the nutrients they need. And it's strength in numbers. They do get some protection by all living together. Now, on the picture up here, this is showing an example of a biofilm that is forming on your teeth. And in lab next week, depends when you're listening to this, we're going to scrape your teeth and we're going to see some of these bacteria that are forming a biofilm on your teeth right now. So this is showing bacteria in your mouth. And again, they're not going to grow unless they're attached to a surface. They have to find a home. But as one attach, they allow other bacteria to attach. Then they form this nice big covering. They start to secrete kind of like a sticky matrix. That's kind of some of that plaque that you scrape off when you brush your teeth. And then as it sits there, that matrix can harden into plaque. That's when your dental hygienist has to chip it off. But as they're all living together, they are feeding together. It's strength in numbers. They're not getting rinsed off by water or food anymore. They literally have this like sticky conglomerate on your teeth. Now, it becomes an issue in the medical field. One, because they can cause cavities if it's on your teeth, but they can form on lots of different surfaces. They can form on medical devices. They can even form in different parts of the body. How they find each other, they do send out a chemical called a quorum sensing molecule that attracts other bacteria to them. Now, we're looking for ways to prevent biofilm formation. If you can prevent bacteria from sticking to each other and sending out these chemicals and attracting other bacteria, you can prevent a lot of different diseases, including cavities. I'm going to see if I found it. A study just came out. They just developed a new toothpaste that can prevent biofilms, which is going to be a game changer in stopping and slowing down cavities. Now, culturing microorganisms, this is like exactly what we would love a plate to look like. So it really means growing organisms, and we're already doing that in lab. It's just taking an inoculum, that's an initial sample, and introduce it into a medium, meaning some type of media, whether it's a plate or a tube. Where the specimens come from that we're going to grow can be environmental, clinical, or stored. An environmental specimen means we go out and just leave our house and we go sample some pond water or some dirt or do an air sample, and we want to find out what's growing. A clinical specimen means it comes from a patient. And a stored specimen means it could have come from the environment or a clinical setting at some point, but now we've grown it in a lab and we keep it stored in a lab. And these are what we're working with. So in lab, if I give you a tube of E. coli or Staphylococcus epidermidis, it's because we had that stored, kept in a freezer, and that's what we're working with in our lab. So if you hear the word culture, that just means it's just another word for saying we're growing organisms in our lab. Pure cultures just means the bacteria are all of one kind of bacteria. They're all one species. And really what we're looking for in pure cultures are those isolated colonies. Again, another word for colony is a colony forming unit. It just means that colony all came from one single progenitor or one parent cell. So one bacteria landed. Anytime we're growing organisms in a lab, we're doing aseptic technique. That just means we're trying to prevent any contamination or at least limit contamination. Two ways that we can try to get isolated colonies. Shoot, that little video's not there anymore. It's just a video of us streaking plates, but you guys have already done that in lab. So two common isolation techniques so we can see what those isolated colonies look like is streak plates, exactly what we just did in lab this last week. So we can try to get isolated colonies. We can see even how many different kinds of bacteria. On this one picture, it looks like we've got red, white, and yellow ones. We can see exactly how big they get when they're not competing. You can see how small they are when they're competing for resources and how big they are when they're not. Another way we can do that is called a pour plate method. We don't do it because it takes a lot of plates, it takes a lot of tubes. It's just kind of putsy. But if we had that initial sample, such as the tube that was on your desk in lab this week, and which we know there's no way we're going to get isolated colonies out of it, but what if we started diluting it, like 1 to 10 dilutions into other tubes? And then we would take a little bit, a small sample from each of those diluted tubes and put it on multiple plates. At one point, we're going to be able to get a dilution level enough that you're going to get isolated colonies. We can do this if we don't think a bacteria can handle the streaking method, but it goes through a lot of tubes and it goes through a lot of plates. So streaking a plate for isolated colonies, much more common way of trying to get those isolated colonies. Now, there are lots of different types of media that we could use for growing organisms in the lab. Most of them are going to be agar-based, which means it's going to have that thick agent to it versus a broth, a liquid. So lots of different choices we could use in the lab. Again, commonly we just use our blood agar, but we'll use some other ones this semester. But my note, most prokaryotes have not been grown in some type of culture media. Although we have lots of choices, we give them every nutrient we think they possibly need. We make them warm, we give them the right oxygen requirement. There are still so many organisms out there that we just have yet to figure out the right combination to grow them effectively in lab. Other ways we can grow them, instead of just on plates, there are going to be slant tubes that we're going to use this semester, which means the agar gets poured in here, but before it cools and hardens, we just tip the tube a little bit. And the reason for that is that it has an area, the slant area, that has a huge surface area for oxygen. And so if a bacteria likes oxygen, you're going to see growth in this area. But it also has this butt region of the tube where there's little to no oxygen. And so if there's an organism that doesn't require oxygen, you're going to see it only growing down in the bottom of the tube. And a lot of ours have different colors to them because where the bacteria grows is where you'll see color changes occurring. Some different terms for different types of media that we use. There's something called a defined media. Defined means we know the exact chemical makeup of what's in that plate or broth. Meaning I know exactly how much of every sugar. I know exactly of how much every element is in there. Now, we actually don't use that many defined medias because bacteria are tricky. And sometimes it's not an exact combination and ratio of certain type of chemicals, but we know some nutrients that they need. We know that they might need a big protein, but we also know it's not just the protein they need the amino acids that are in the protein. And so more commonly, we use what's known as complex media. So in complex media, there are nutrients that we put in there that start to break down. And a lot of them are proteins. Example, we can put milk proteins, soy proteins, beef proteins that we know are going to start to break down while they're in the media. And because they're proteins, they break down into basic amino acids. And we know the bacteria are going to be using those amino acids as the proteins are breaking down. But because we don't know exactly where it is in the breakdown process, we don't have an exact chemical makeup for it. We also will use some media this semester in lab that's known as selective, which is going to be on some of my next slides. I've got an example, some that are differential, as well as what are just known as transport media. So selective media, and I'll talk about it in lab as well, it has some substance that inhibits the growth of one type or one group of bacteria, but it has no effect on another type or group of bacteria. Example, this is a nutrient agar that if I went outside and just swabbed the cement, this is probably what my plate's gonna look like. But there's so many bacteria, there's no isolated colonies, they're all competing for space and nutrients, and I don't really see what's going on. But what if I want to know if there's fungus growing on that cement sample? Well, then we can use a plate. It's a selective plate. This is an example. It's called Sauerkraut agar. It has antibiotics in it. So we know there's going to be bacteria on the cement. And I don't want to see the bacteria. So the antibiotics will inhibit the bacteria, but they will have no effect on the fungus. And so we can truly see what's going on. Another example is differential. And this means you're going to see a difference in colors based on whether the bacteria can do something with some type of substrate. So we're going to give the bacteria substrate. It's usually a sugar. and different kinds of sugars. And we're just looking to see, can the bacteria do something with that sugar? If it can, we're going to see a color change. And that keys us in to what the bacteria is, because not all bacteria can break down certain sugars. Example of a differential media actually is a blood agar. Because of the red blood cells that are in there, whether the bacteria can break it down fully, that you could actually see right through it, or partially, where it looks a little greenish, or they can't break it down at all, that's a huge clue. And I'm like, the plate's going to look different. It's going to look like there's some clearing, it might look greenish colored, or it's going to look like no change whatsoever. So we're going to see some differences in colors. This is showing another plate we're going to work with this semester called the Microbial agar, which is both selective and differential. So this is showing nutrients going to grow anything and everything. So we put 2 bacteria, E. coli and Staph aureus on here. Both are going to grow. It's meant to grow anything. But a Microbial agar is selective. It will only grow gram-negative bacteria. So Staph aureus, which is gram-positive, is not going to grow. But E. coli is happily growing. So it's selective. It's got an inhibitor to inhibit gram-positives from growing. But it's also differential. Because now what if I put 2 different gram-negative bacteria on here? They're both going to grow. Because they're both gram-negative, they're not inhibited. But we notice some differences in colors. And on this particular plate, what we're looking for is, do we see pinkish purple colonies or not? Although this turns a yellowish, that's not what we're looking for substrate breakdown. We're looking, do we see pinkish purple colonies or not? So we can see now differences in color. Huge way, super easy way to tell us, a little bit more about what kind of bacteria it is. Sometimes differential media is not just on plates. We have some tubes as well where we give it some substrate, usually a sugar, and we can look to see if we've got color changes or not, or even if it produces gas when it breaks down that substrate. And then there's transport media, and we'll look at some examples of this up in lab. Again, transport media, if you collect a sample from a patient, you want to preserve that bacteria until it gets to the lab for testing. Again, the faster you can get it there, the better the results are going to be. But you're not just going to take a swab and walk down the hall. You have to put it in something. And so we call that transport media. Sometimes it's in a liquid, sometimes it's in a tube. If we suspect an anaerobic organism, there are certain vials that will keep the air in there anaerobic. So we have to put it in something to get it to the lab. And again, the faster you can get it there, the better results you're going to have. And as I mentioned before, there's a lot of organisms out there that we just have not been able to grow in the lab using regular agar and media techniques. So we've got these kind of unique ways we can make organisms grow. One is animal and cells. So we know that there are some microbes that can't grow on the media, so we can grow them in different animals. An example, the bacteria that causes leprosy and even syphilis, we can just not grow those things in the lab. We just have not been able to figure out any kind of media or environment, but we know they grow really well in armadillos. And so if you have to grow that particular bacteria for research or something else, you're probably going to have to get some armadillos in your lab. It's also another reason, if you are anywhere traveling where there are armadillos, don't play with them because they can easily be carriers of both leprosy and syphilis. Random factoid. Excuse me. Cell cultures. We know there's one organism, viruses, that require cells to grow. So if we want to grow viruses in a lab, we first have to grow cells in the lab and then inject the virus, or at least expose the viruses to the cells, and then the cells become virus-producing factories. We also have what's known as enrichment culture, which means it's going to have some type of selective media. So we kind of want to narrow down what we want to grow. But then we're going to add a large amount of certain type of limiting factor that one organism needs. So it's enriched because we're adding a lot of some type of limiting factor just to optimize a very specific organism to grow. We also know that some bacteria don't like to grow with lots of oxygen present. I mean, if they like oxygen, we can expose them just to the air. But some like a low oxygen culture. Again, they would like a little less oxygen to be happy. And we'll do this in lab this semester, is that we can grow them in a candle jar. We can put our plates in there. We're going to put a candle in there. We're going to close the lid, and the flame's going to go out. Now, the reason why the flame goes out is that the flame was consuming some, not all, but some of the oxygen until there wasn't enough oxygen, and then it went out. That now produces a low oxygen environment. There's also going to be some organisms, not that we're going to work with this semester, that require zero oxygen. So they're completely anaerobic organisms. If we suspect a patient has one of these, we do have, they're called gas pack jars. And we've got an example in our lab that we can put the plates in there. We open that little packet up. It releases hydrogen. And it's going to take all the oxygen that's found in this little canister and it's going to bind it up as water. And so there will be no more oxygen gas present because all of the oxygen is bound up in water and it puts it into that zero oxygen anaerobic environment. Now, we kind of talked about how we want to grow these different organisms, given them everything they want, but what if we want to just preserve them? Well, if we want to preserve them for just a short period of time, we can put them in the refrigerator. That's why we have one in lab. If we want to preserve them for a lot longer, we use a deep freezer. This would be like a minus 80 degrees Celsius freezer. Pretty much puts these in like a coma, and they're going to stay there for years. If we want to keep them even longer, for research, for whatever, this is usually like the CDC, it's lyophilization, which is freeze drying it, which can preserve them for decades. Now, if we do make our bacteria as happy as can be, they grow by binary fission. Just means they start as one, they double everything, including their DNA, and they split into two. And that's it. And then they're going to do that all over again. And if you make them super happy, they can do this whole process in less than 20 minutes. So a generation time is how long does it actually take for one bacteria to grow and divide. So it's also called the doubling time. So this is showing if this bacteria's generation time was 30 minutes, after 30 minutes, one bacteria is now 2. Well, if we give those 2 bacteria another 30 minutes, so now we're 60 minutes in, those 2 bacteria now become 4. After another 30 minutes, they become 8. After another 30 minutes, they become 16. After another, we just keep doubling. This doubling every generation time is called a logarithmic growth, where it's going to start real slow and then it's going to go real fast. Most organisms out in the world grow what's known as arrhythmically, which is more just a slow, steady growth, not bacteria. You make them happy, they are going to grow very fast, constantly doubling. Now, as soon as you put a bacteria on a plate, again, we give them those organisms, all their nutrients, we put them in the incubator, make them happy. They are going to do logarithmic growth, that exponential, super fast growth, but not the second you put them on the plate. There is what's known as a lag phase. It just means the bacteria, it takes a little bit. They might have to produce some enzymes to break apart some of the nutrients you gave them. And so there's a little bit of a lag phase. And then as soon as they've got everything ready to go, they just multiply just super fast and you get high numbers very quickly. Now, it's at this point is where we generally pull them out of the incubator when they're in this really happy growing time. Because at this point, you see lots of bacteria and lots of growth. So they're great from gram staining. But at some point, you're going to get to what's known as a stationary phase. Some of the nutrients are going to start to go away. Some of the waste products are going to start to accumulate, and you're going to start to get dead cells. And dead cells don't gram stain well. So this is where we try to pull them out. We don't let them sit in the incubator longer than 24 hours because we really want to see lots of living bacteria. Doesn't mean they're not still growing, but pretty much for every bacteria that grows, there's another one dying. And then at some point they get into an area, it's called the death or decline phase, because there's more cells dying than are being produced. This is, again, just another illustration showing that lag phase, that super fast exponential growth. Stationary, again, just as many growing as are dying, and then the death phase, where there's more dying than there are growing. But how do we know that they grew? How do we know? So there's different ways we can measure microbial reproduction to know that they were actually reproducing. There's direct methods that don't require any incubation whatsoever, and then indirect methods that do. So direct methods, again, if you've got the money, they're a lot faster. You don't need to incubate them. So they can, sometimes they're putzier, but they can be very, very efficient. There's a couple different electronic devices that we can use. One's called a Coulter counter, which I just like the name of it, that it is showing here that if I had this big test tube and all these little green bacteria floating around in it, I can put this electronic device, it's in its own little machine, and there's literally a little space between where electricity flows. And it can measure any time a bacteria passes through that space because it's disrupting the electrical flow. And so as a bacteria goes through that little space, it disrupts the electrical flow, machine counts it as 1. And it can make a pretty good estimate of how many bacteria are in a tube. A similar one's called flow cytometry, and I say similar because it's got another machine. This uses laser beams, and as you send bacteria through a small tube, there's a laser beam that as it hits a bacteria, the light kind of deflects off the bacteria, and then there's a machine that counts that deflecting off of the bacteria. And it makes a pretty good estimate. These, again, can be very expensive, and we're not going to use them in our lab. We can also do microscopic counts. For us for the semester, we don't need to, but it's actually not that hard. There are some slides that you can purchase that have etched grids right on them, so we reuse these, we don't toss them. And you can put a drop of broth on there, put a cover slip on there, and we know exactly how much fluid is between the slide and the cover slip. So we have a volume that's right in between that cover slip and slide, and underneath the microscope we can physically count the different bacteria that are in a particular grid, and then we go back and do the math of there's this many bacteria per this much, on the grid, and we know the volume that's in that space, and we can say, okay, well, there's 15 bacteria per microliter of fluid. The direct methods require incubation, so they take longer. They can usually require more supplies. And these are my next slides on here of dilutions, filtrations, and most probable number. So there's the serial dilutions. You can already see how lots of media that happens, but it's one way we can do counts. Is if we take our original broth, super concentrated, and we start diluting it. 1 to 10 dilution, another tenth of a dilution, we're at 100, another tenth of a dilution, we're at 1,000, another tenth, we're up to 10,000, and then another one, as we put it on the plate, we can be up to 100,000 dilution. Well, if we know how many times we've diluted it, and even if we don't even put a full mil on a plate, we're only going to put 0.1, which is technically another 10th dilution, we can get to an area and say, well, I've got 6 colonies, and I've got a 10,000 dilution, but because I didn't put a full mil, I only put a tenth of a mil, that's another tenth of a dilution. This is really now technically 100,000 dilution. So 6 colonies per 100,000, that means there's about 600,000 bacteria per milliliter. If I go back a dilution, because this just means too numerous to count, you wouldn't want to count that plate. But if I get to a dilution where I'm like, well, there's 65 colonies, and I go back, well, 65 times 1,000 times another 10, you're at 650,000. So you're getting a pretty good estimate about how many bacteria there are per milliliter. Again, it's putsy. This is a lot of tubes, a lot of plates, and you still have to wait for incubation. A simpler method is called membrane filtration. is you can filter, say, a liter of fluid, and you can catch all the bacteria on a plate, on a grid, grow them up, because you got to make them happy, you got to make them visible, and then you can take a look at it 24 hours later and say, okay, I put a liter of fluid through here, and I caught this many bacteria, I don't know, 70 bacteria. Well, there's 70 bacteria per liter. A lot faster. Again, this is usually used if you have low numbers of bacteria, but a lot of fluid that you got to look at. Most probable number is used when you can't grow the bacteria on any kind of agar, that it has to stay in a liquid form. And so you still do various dilutions, and then you add the different dilutions up to 5 tubes, and you look to see which ones turn colors based on the dilution. So if we notice, the more diluted we get it, the less tubes turn yellow. So we can see that we're getting more and more diluted because less and less of them are doing the color change. Then there's a big most probable number table that you just go back and look at, like based on my dilutions, based on how many tubes turn yellow at each of the different dilutions, there's a table that tells you approximately how many bacteria were in there. Another indirect method that you can do, that doesn't require any incubation, is you can just do metabolic activities. Is if there's a lot of bacteria present, you're going to see sometimes a very significant color change in a certain test. We can look at dry weight. We can filter our organisms, dry them out, and then just weigh them and just get the mass of how many bacteria there are. We can look at genetic methods. How much DNA is there present? The more bacteria, the more DNA. Our easier way is turbidity. How turbid or how cloudy is our broth is approximately how much bacteria there is. So if it looks super cloudy, lots of bacteria. Not very cloudy, not a lot of bacteria. It's not an exact science at all, but at least it gives us some kind of estimate. There are some machines that can look at turbidity to at least give us some numerical range of turbidity. Again, they shine light through the broth. If there's no bacteria present, truly the light just shines right through it, and you're going to have a pretty low number. If there's tons of bacteria, the light is going to keep bouncing off all those bacteria. Not a light goes through, and you end up having a higher number because the light is getting absorbed. So just some different ways, again, we can look to see, did bacteria grow? Are they there? Because that's what we're going to be doing a lot in lab, is growing bacteria. And sometimes we want to know that bacteria is there and present for us to work with.